As we already differentiated between plant cell and animal cell, let's discuss some of the organelles which are present only in plant cells like plastids and outermost covering of the plant cell, which is cell wall. Coming to the plastids, these are found in all plant cells and euglenoids. Depending on its color and types of pigments present in them, these are divided into three types: leucoplast, chromoplast and chloroplast. Coming to the leucoplast, these are colorless plastids which are present in non-green cells. These are of three types: amyloplast, which contains starch, example potato tuber; alluloplast, which stores oils and fats; alluroplast, which stores proteins in them. Coming to the chloroplast, these chromoplasts, which consists of fat-soluble carotenoid pigments like keratin, xanthophyll, etc. These produces colors like yellow, orange or reddish orange to the ripening fruits, because here the chloroplasts are transformed into chromoplasts, which gives colors to the ripening fruit. Coming to the chloroplast, these are lens-shaped oval or spherical ribbon-like structures which are present in the mesophyll cells of the leaves. These are greenish plastids which are present in green cells of the leaves. These contain chlorophyll and carotenoid pigments which are responsible for trapping light energy, which is essential for performing photosynthesis. These are about 5 to 10 micrometer in length, 2 to 4 micrometer in width. There are variable number of chloroplasts are present in cell, like in chlamydomonas, which are present 1 per cell, and in green algae, these are present 20 to 40 per cell. Let's discuss structure of chloroplast with the help of diagram. Here, the chloroplast is double membrane organelle which consists of two membranes, which is outer membrane and inner membrane. Between outer and inner membrane, it consists of space known as intermembrane space. Inside the inner membrane, the fluid-filled space known as stroma, which contains number of flattened membranous sacs known as thylakoids, and these thylakoids are arranged in stack-like piles of coins known as grana, singular, which is known as granum, and these grana are also known as integral thylakoids. And thylakoids of different grana are connected with help of tubules known as stoma lamellae. These consist of 70S ribosomes, which are smaller than the cytoplasmic ribosomes. And these chloroplasts, as we already discussed, these consist of pigments like chlorophyll, which trap light energy essential for photosynthesis. These perform photosynthesis, light reaction in the thylakoids and dark reaction in the stroma. These also contain starch in them. These mitochondria and chloroplasts are semi-autonomous organelles because they are capable of self-duplication, as they have their own nucleic acid like double-stranded DNA and RNA molecules, 70S ribosomes, and it also has ETS and ATP forming machinery in it. Let's discuss about cell wall. It is non-living rigid structure which is found in fungi, algae and plant cell. Here in fungi it is composed of chitin, in algae it is composed of cellulose, galactans and some of the minerals like calcium carbonate, and in plant cell it is composed of cellulose, hemicellulose, pectin and proteins. Based on age of the plant cell, in young plant cell it forms a single layer which is thin, elastic and capable of growth, which is known as a primary cell wall, and in mature plant cells, inside to the primary cell wall it forms as secondary cell wall. Coming to the middle lamella, it is the connecting material which holds the adjacent cells together, as you can see in the picture. Here, the cell wall and middle lamella are transversed by plasmodesmata, which means these plasmodesmata, which extends or passes across the cell wall and middle lamella and connects the cytoplasm of neighboring cells, and this allows transport of water, nutrients, and provides cell to cell communication between the plant tissues. Coming to the functions of the cell wall, it maintains shape of cells and protects cell from mechanical damage and infection, and it provides cell to cell interaction, and it provides barrier for undesirable micromolecules. Let's discuss about vacuole. It is present in both plant and animal cells, but in plant cell it occupies large volume up to 90% volume of the cell. Let's discuss structure of vacuole. It is single membrane bound organelle in the cytoplasm. It contains water, sap, excretory products, and waste materials. The single membrane, which is known as tonoplast, it facilitates transport of number of ions and materials against the concentration gradient into the vacuole. There are many types of vacuoles, like contractile vacuole, which is found in amoeba. This helps in osmoregulation and excretion, and foot vacuole, which is present in protists, which helps in engulfing the food particles, and gas vacuole, as we already discussed about it in prokaryotic cell. These are found in prokaryotes, which provides buoyancy to the cells. As we already discussed about various cell organelles of eukaryotic cell, its structure and function in detail, here we are going to cover about nucleus, which is the largest cell organelle and controls all the activities of the cell. It was first described by scientist Robert Brown in the year 1831, and based on number of nucleus present in a cell. If the cell contains two nuclei, then it is known as binucleate cell, as in case of paramecium. If cell contains many nuclei, then it is known as multinucleate cell. And if cell lack nucleus, as in case of mature mammalian RPC and sieve-tube cells of vascular plants, then it is known as anucleate cell. Let's discuss about structure of nucleus in detail. At non-dividing phase of the cell, which is known as interphase nucleus, has nucleolus, chromatin, nuclear membrane, perinuclear space, and nuclear pore. Let's discuss about each and every part of the nucleus in detail. Coming to the nuclear envelope, which is the membrane that separates nucleus from the cytoplasm, is differentiated into outer membrane, which continues with the rough endoplasmic reticulum, and inner membrane, which is smooth. Between these two membranes, there is a space which is known as perinuclear space, and there are nuclear pores which are formed by fission of these two membranes, and this forms as passage of substances to move inside and outside the cell. And there is transparent fluid which fills the nucleus, which is known as nucleoplasm, that contains nucleolus and chromatin material. Coming to the nucleolus, this is non-membrane bound spherical structure. This is a site for ribosomal RNA synthesis, and there are large number of nucleolus present in a cell, and that indicates that this cell is involved in protein synthesis. If there are large number of nucleolus in a cell, that indicates the cell is actively involved in protein synthesis. Coming to the chromatin material, it's a Greek word where chroma means color. It is the highly extended and elaborate nucleoprotein fibers. Here, it is named by scientist Fleming based on its ability to stain with some of the basic dyes. And this chromatin material, which is composed of DNA molecules, some of the basic proteins like histones, non-histone proteins, and RNA molecules. In human cell, the DNA, which is 2 meters long thread-like structure, which is distributed among 23 pairs of chromosomes. at different stages of cell division, where we are going to discuss in chapter 10 cell cycle and cell division in detail. The chromatin material is condensed to form chromosomes. Let's discuss about structure of chromosome in detail. Here the chromosome consists of two identical halves which are known as chromatids, and these chromatids held together at a point which is known as centromere, and this centromere appears as narrow region which is known as primary constriction, and sides of these centromere there is a disc shaped structures which are known as kinetocodes. Based on position of this centromere, there are 4 types of chromosomes. Let's discuss about each and every type of chromosome in detail. Metacentric chromosomes: if the centromere is present at the center of the chromosome and divides into 2 equal arms. If the centromere is present slightly away from the center of the chromosome, then it divides the chromosome into one shorter and one longer arm. Then it is known as submetacentric chromosome. If the centromere is situated close to its end and divides into one extremely short and one extremely long arm, then it is known as acrocentric chromosome. If the centromere is present at terminal portion, then it appears as single arm. It is known as telocentric chromosome. And few chromosomes has additional constrictions at its end, which is known as secondary constrictions, and which is present beyond to the secondary constrictions as small fragments. These are known as satellites. If you like this video, don't forget to like, share and subscribe to my YouTube channel. We'll meet again in our next video.